Genetics research has benefited greatly from the release of the human genome sequence. Pyrosequencing is a unique system that allows the analysis of genetic variation as well as RNA allelic imbalance, DNA methylation status, and gene copy number. In this video, we demonstrate a basic pyrosequencing reaction for genetic analysis. Hi, I'm Cristi King from the laboratory of Dr. Charles Eby and Dr. Brian Gage in the Department of Internal Medicine at Washington University School of Medicine. Today we are going to show you a procedure for pyrosequencing. This procedure is useful because it's one of the most thorough yet simple methods used to analyze SNPs. Pyrosequencing is based on sequencing by synthesis, taking advantage of the release of pyrophosphate whenever a nucleotide is incorporated in an open 3' DNA strand. Once a plate is loaded, nucleotides are incorporated based upon a sequence provided by the software. Once released, the pyrophosphate is used in a reaction that results in the release of ATP, which is used by luciferase to convert luciferin to oxyluciferin, resulting in the emission of light. The emitted light is collected by a CCD camera and recorded as peaks, also known as pyrograms. Any nucleotides not incorporated into the DNA strand are degraded by apyrase to prevent background noise. This procedure involves the following steps: assay design,